A very big hello to my wonderful PU2 champions. I hope all my dear children are doing super great. Yes, so our practical exams are going to soon hit us, and to be safe from that big hit, we need to have some practical tips, and I'm going to discuss about that in this particular video. But before I go any further, I want one thing from you guys, and that is please put down your biology practical examination dates in the comment section below, because we are planning something for you, and if at all you let us know your plan, we will be able to plan accordingly for you guys. Okay, so that is the reason. Please put it down in the comment section. Now going ahead, how many marks does the practical give you? It gives you 30 marks. Biology paper is of how many marks? 100 marks, ma'am. So 30 marks comes from your practicals, rest 70 marks come from the theory paper that you will be giving. Now, one thing that you need to keep in your mind is that the 30 marks also have some bifurcation. Suppose, if you do all the experiments properly and if you write everything which is needed in the practical examination, you will get 20 marks. Now, if at all you make sure that you have attended the theory and practical classes properly, you will get 5 marks. Now, that is something that is under your control, but it is already over, so now you cannot do anything about it. Okay, that is there. But there is something that you can still do, and that is you have a complete and duly certified practical record. If at all your practical record is complete and duly certified, what is going to happen? You will get the 5 marks again. In that particular case, your total marks would be 30. There is no viva this time, which is why you can be really happy as well. So a happy news is there, and also sad news is there. That is, as I said, if your attendance does not match properly, your marks over here is also going to vary. So have that in your head and talk to your teachers. Yes. Okay, I know that won't be changing much things, but yeah. Now, talking about the question paper, how does it look? So my dear children, you have 4 experiments in the first question. What? We have to do 4 experiments? No. So basically it's all options given to you, but you cannot pick any options like that. There is lottery system, there will be chits given to you and you have to pick it up. Whatever experiments come up, you have to do it. But that means that we have to be sure about all these 4 experiments, because any of it can be asked to attempt, and all of it is of what? 5 marks. So one is pollen germination experiment, one is pollen tube growth experiment, and one is transverse section of ovary, where you cut the ovary and look for the placentation in the case of plant cell. And then there is what? A nucleic acid staining experiment, where you say what is the shape of the cell and what is basically the number of nuclei observed inside the cell. So these are the 4 experiments that you have in question number 1. Any one of them has to be attempted. The choice is not yours. Basically, it depends upon your luck. Now, talking about question number 2, this is common for everyone, compulsory for everyone for you to do, and that is create a temporary slide of what? Prepare a temporary slide of what? The given material, which is onion root tip, and over here you have to identify one stage of mitosis and also put down the comment that is observed, as in put down the stage which is being observed. So one stage you identify, you have to write about that particular stage. So there is metaphase, prophase, telophase, anaphase, whatever phase it is, identify it properly and then write about it. Talking about it gives you marks. I'll be talking about all of it in detail, do not be worried as well. Okay, and then there is a pedigree chart question. You have to identify the pedigree chart. If you identify it properly and you write the comments properly, you get the 3 marks. Then there are 3 more things which have to be identified and commented upon. This gives you 3 and 2 and 2 marks. Okay, so now let's slowly get into one by one each of these experiments. So question number 1 experiments, the first experiment we all know, it is what? Pollen germination experiment. So over here I am not going to tell you the steps which are there in pollen germination experiment. Why? Because preparation of the slide, if I talk about it, I might say something else. The teacher who might take your examination might have some other opinion onto it, and if you follow my things, you might not get marks there. Okay, so depending upon school to school, things vary, certain stains vary, so I'm not talking much about it. But as I said, if at all you give us the dates, probably we will show the experiments. That's exactly why we were asking for the dates. We can show you the experiments and you can ask to your teachers if at all this are the steps that need to be followed or not. And if not, then you can make changes accordingly. But at least you will have an idea. Okay, so preparation of the slide for this particular thing gives you 3 marks. If you do it properly, you get the 3 marks. Now, there are 5 marks in total, right? 3 is done. Now what about the 2 other marks out there? We need to discuss about that, and that is easy to grab marks. Listen to me. So over here, the flower that you will usually take is what? Visca rosae, okay? And you will shed down the pollen grains on the glass slide like this, and the pollen grains are somewhere over here, something like this. Imagine. Now what do I do to do? I need to segregate this. Suppose I make a segregation like this. So this is 1, this is 2, and this is the third column. And over here there is table for calculation. If I make this table properly and if I put the content down properly, what I'm going to get? I am going to get 1 marks. So what is there in the table? Let's see that. So over here we need to have all your attention. So number of observation as a number of columns made 1, 2, 3. Now total number of pollen observed in each of the column, that is denoted by capital N. So ma'am, what is a capital N over here? Capital N value in column 1, 1, 2, 3, 4, 5. 5 pollen grains are there, so you write 5 over here. You got it? Now out of this 5, how many number of the pollen grains are germinated? You have to talk about that. So what do you think? How many of them are germinated? Wait, okay? I'll show. So suppose this has made the pollen tube, this has also made the pollen tube. And now I show you. Just a second. Yeah. So out of 5, 2 of them have made the pollen tube. This is known as germination. Pollen tube formation, my dear children, when it happens, you call this process as what? Germination. So how many of them have germinated? 2. So out of 5, you have to write 5 over here, and then you have to write 2 over here. Suppose nothing has germinated, you write 0 over there. But you have to write something out there in that particular box. It's not like there is no germination, so you don't write anything. Don't do that. Okay? And then make a sum total of all of it over here and put it down here. Similarly, make a sum total of all of it and put it down over here. Got it? Now then, that gave you what? 1 mark. What about the rest 1 mark out there? So you have to find the percentage of pollen germination. This formula again gives you half marks. And not just this formula, you have to replace the content over here in this particular formula. So you did the total of this n, right? So small n is equals to what? You have to write it. So small n is equals to x, whatever number is there. Capital N is equals to y. Mention that and then replace it in this particular formula. Write the formula first and then write this and then do the whole replacement. This gives you half marks. Then calculation of the pollen germination will also give you what? Half marks. Write it properly, you'll get those marks. So 2 marks easily you can grab. Okay, preparation of the slide can be a little, tricky for you guys, but you will be able to do it. Don't worry. Okay, do not worry about it. So I hope this is clear. Okay, so anyone who is watching the video, go through all this thing. Do not forget capital N denotes what, small n denotes what, and write it properly and then only go. Fine. This is pollen germination experiment. Now what do we have? Okay, and this is how it looks. Germinated pollen tube, my dear children, it looks like this. Pollen grain looks like this. Fine. Now we have pollen tube growth. So pollen tube growth is basically, again, we have to see how the pollen tube is growing out there. Now this is again an easy experiment. You take the visca rosae and then you cut it with a needle out there. So what are you going to get? Where is the pollen tube going to grow, my dear children? So on the stigma it grows. So this is a stigma, this is a style, and over here you would be having the pollen grain. Some of it is going to grow out there forming the pollen tube. So this is the thing that you need to do. Okay, for that again you need to do the preparation of the slide. That will give you 3 marks if you do it properly. There are steps involved, there is heating involved, there is staining involved. So it's not like a one-step process, I am telling you again. And the diagram with 4 labelings, if at all you make this, you are going to get 2 marks. How? For each correct labeling you get half marks, and there are 4 of them. So 4 into half is equals to what? 2 marks, my dear children. So what are the labelings that you should be doing? Pollen grain should be labeled, it's there on the stigma. Stigma should be labeled, pollen tube should be labeled, and style should be labeled. Now you tell me, if at all you were someone who didn't know that writing this also gives you marks, aren't you happy now? Yes, you are happy, right? If at all you are happy, what are you doing? Subscribe to the channel right now, because if at all you want much more contents like this related to biology and all the other subjects, you need to subscribe. Otherwise, how will you get updated with it? Right? So do that right now. Okay? So I hope this part is clear, my dear children. Do not do any mistakes, and if you have doubts, put it down in the comment section. Fine. Now there is transverse section of ovary. Transverse section of ovary. What do you mean by that? So there is stigma, there is style, and there is ovary over here. This is China rose, or you also call it as hibiscus, right? Again with a needle you open it and you basically take the style and the ovary outside. Okay, you take the ovary outside. It looks something like this. So style is this and the ovary is this. Now transverse section means you're cutting like this and you're viewing it like this. You're not cutting it like this, rather you're cutting it like this and seeing what's happening inside. Okay, so again in this particular thing, preparation of the slide gives you 3 marks. Whereas reporting the number of locules in the ovary give you 1 marks. Ma'am, what do you mean by that? Listen to me carefully. So over here I have the ovary, right? Usually inside the ovary, what do you have? You have the ovule, and ovule is supported with the help of placenta, as in placenta gives the initial nutrition. Fine. And this space which you have in the ovary, it is known as locule. It is known as locule. But this is not exactly how every plant's ovary looks. Okay, this is not exactly how every plant's ovary looks. It is different. So in the case of China rose, the ovary looks something like this. If at all you make a cut and see, it looks something like this. So over here, one. Just give me a second. Okay. So over here, we have the placenta. What do we have? This is the placenta. Placenta is in the middle. This type of placentation, where the placenta is in the middle and basically it's forming ridges in between. In the ovary, you call this as axial placentation. And over here, in this particular placentation, all your ovules are going to be around the placenta like this. You would be having 2 ovules over here. So this space that you see, this one, this one, this one, this one, this one, they are all locules. But the locules are divided into what segments? You will now call this as pentalocular condition. What do you call this? Pentalocular condition. Penta means 5. So over here in the case of China rose, you have 5 locules, 1, 2, 3, 4, 5. And each of them, my dear children, would be having 2 ovules. Ovules are supported by the placenta. The placenta is in the middle, and this placenta is also dividing the locule into what? Pentalocular condition. I hope you got it. So reporting the number of locules in the ovary gives you 1 mark. So it is pentalocular. If you write this, you will get 1 marks. Now reporting the type of placentation will be giving you another 1 mark. So what is the type of placentation? It is axial placentation. Axial placentation. So depending on the position of placenta and how it is there in the ovary, there are different types of placentation. So over here I have a picture. When it is like this, as I told, in the middle it's there and it is having ridges, it is known as axial placentation. If in the middle it is there but there are no ridges, you will call this as free central placentation. You might have studied in your 11th grade. And if it is towards the, what do you call, periphery over there, you call this as parietal placentation. If it is on one side only, you will call this as marginal placentation. If it is over here, you will call this as basal placentation. So over here, you need not worry. China rose is there, and China rose, there is what? Axial placentation that you need to remember. So over here, I have written one thing. No marks for the diagram, but please draw it. Even though there is no marks for the diagram, but please draw it. It is going to be really good. And report the type of placentation as well. Okay, works? Okay, fine. So 1 mark you will get from that. Fine? Fine. Now going ahead. So total you have got 5 marks from this. Going ahead with the next thing, that is nucleic acid staining experiment. Now what is happening over here? You are going to take the onion peel. Onion peel is taken, the innermost peel, it is very light peel, you're going to take it, and you, with the help of that, you're going to prepare the slide. Preparation of the slide will give you 3 marks. Now reporting the shape of the cell is going to give you 1 marks. So basically, it is considered to be a rectangle. It's a plant cell only. It is considered to be rectangular in shape. Not exactly rectangular, but yes, we say so. But you can definitely confirm with your teacher who is going to take the practical examination experiment also. Just have that confirmation. But know that writing this gives you 1 mark. So you should not miss on that 1 mark. No, so please know that in, in advance. Now, reporting the number of nuclei will also give you 1 mark. So over here, if this is a cell, this region which is darkly stained, it is known as what? The nuclei. It is the nuclei. So this nucleic acid staining experiment, right? Your nucleic acid, which is a DNA, my dear children, is negatively charged. So you have to use some positively charged stain to view up the negatively charged DNA content out there. So the number of nuclei, as you can see, all of them is just having 1 nuclei per cell. So I'll say it's uninucleate in condition. It is what? Uninucleate in condition. And shape, as I told, it's rectangular, but still you can talk about it to your teacher in advance. Okay, and that is how you're going to get your 5 marks in this particular experiment. Now, Going ahead, question number 2, experiment. As I said, it's common for everyone. Preparation of the slide gives you 3 marks. What is this experiment on? Forming the temporary mount of onion root tip to study mitosis. So you take the onion root tip and you study the mitosis. For preparation of the slide gives you 3 marks. Now if you identify any one of this stage, it gives you 1 mark for identification. After identification, you also have to comment on it. So whatever stage you have identified, for example, metaphase is something that you have identified. So you have to say that all the chromosomes are lined up over here in the metaphasic plate or in the equatorial plate. So all of that you have to mention some features of that. So any 2 stage, commenting on the stage observed. So any 2 stages, not 2 stages, I think 2 points would be there, and for that you have to write, for that you will get 1 marks. So identify one of the stage and also comment on the stage observed, you will get what? 1 marks. So any 2 points is what is meant. Okay, any 2 points about that stage. There is prophase, metaphase, anaphase and telophase. So in prophase it's all together. In metaphase basically it's in the middle on the equatorial plane, and then in anaphase they start separating. In telophase they have completely separated at the terminal position that they reach. So you should know that. Okay, identify, you should be able to identify it properly. Fine. Now if, as I said, if at all you want me to help you with that, let me know in the comment section below. Okay, got it? Yes. Fine. Now everyone who is preparing for KCET, one very important information for you guys. We have already launched the ABHYAS KCET test series, and it's high time to take it up and start practicing your questions, because there is chapter-wise PYQs as well that we have newly added. So once you're done with your PU2 chapters, go solve some PYQ questions of your KCET, and with a limited time also, because all of it is under real exam like pattern and conditioning, so that you get an idea about your speed and accuracy also. Later on you can go ahead and give the mock test. There are 35 KCET mock tests, and there is PCMB coverage. So usually biology is not there, but this time biology is there. So enjoy it. Previous time also it was there, but this time we have launched it in a proper sense. So previous time we launched it a bit late, the biology part, but over here you have got it ready. So take it up. Okay, and the pricing for this is rupees 1499. If at all you wish to take it up, the link will be given in the description section. My idea would be to definitely please, you should be going ahead and consider it and to take it up, because it is a really good series. You can see that people out of 3 lakh children, our children got the ranks 171, 443, 693, and that's not at all a small deal. So that is the reason I'm not over-exaggerating things and not telling too much about it. Please go take it up if at all you think it works for you. Yes? Now question number 3 is on the basis of what? Identification of any one of the given below pedigree chart. So that identification will give you 1 marks. Okay, commenting on it, giving any 2 points on it will be giving you 2 marks. So autosomal linked dominant trait, autosome linked recessive trait, X-linked dominant trait, X-linked recessive trait, and Y chromosome linked trait is going to be asked. Okay, so the pedigree charts can be multiple types. Pictures would be given to you. You have to identify, you have to write it down. So how do you identify it? That is a very important question. So as I said, I was planning to make a separate video on pedigree, because it is useful for your PU2 also. Let me know if you need it really fast. But yes. So these are the 4 things. Have this in your head. 5 things, sorry. Have this in your head. I'll explain you one very important thing right over here. Then if you think the explanation is fine, you can let me know. Okay. Then I'll explain more. So there is inheritance of autosome linked dominant trait. So autosome means it is not your sex chromosomes which are being affected. It is the other chromosomes which are affected. So we have 46 chromosomes in total. Out of 46, 44 of them are autosomes. 45th and 46th chromosomes are sex chromosomes. Either it can be XX or XY. If at all you have any genes which are affected on the X chromosome, you call this as X-linked disorders. But it's not like that. Here, the autosomes, the genes on the autosomes are getting affected, which is why I'll call this autosomal disorders. Now there is autosome linked dominant disorder. Okay, autosome linked dominant disorder. First of all, how do I understand whether a disorder is dominant? So if at all I say that in every single generation a person is affected, I will call this as what? Dominant disorder. Okay, if at all I see skipping of generation, one generation is there, another generation is not there, I'll say it is recessive disorder. But here I can see the mother is affected, the son and the daughter is affected. In the next generation, my dear children, their daughter is affected. In next generation, their daughter and son is affected. Someone is affected in every generation. That is why I'll call it dominant. Now, why will I call it autosome linked? Because over here, if at all you see the pattern, the lady is affected, the son and the girl child, both of them are affected. Again, the girl child is affected. Here again, the girl and the boy child is affected. Are you able to come to a conclusion that any sex is being affected more? No. So we say it is autosome linked dominant trait. It's dominant, we already know, and we know it's not sex linked, because if it was sex linked, my dear children, you will see a different pattern altogether. For example, if I come over here, this is sex linked or X linked dominant trait, and over here you are going to see something unique. For dominant, you know, it has to be there in every generation, so it is there in every generation. But how do we understand whether it is X linked? Here, my dear children, you can see father to daughter transmission. If the father is affected, daughter is definitely going to be affected. In X linked dominant trait, if the father is affected, the daughter will definitely be affected. So here the father is not mating, so you cannot see anything. The boy is not mating, but here you can clearly see the boy has mated and they have daughters, and the daughters are affected. Why is that so, ma'am? It's just an easy understanding. There's a trick. As I told you, from the father, it's always the daughter who is going to be affected over here. There's a crisscross pattern that you're going to see. So listen to me carefully. Why? So it's an X-linked dominant disorder. That means if your one X chromosome is affected, you will have the disease, right? So this man's one X chromosome is affected. The females, none of the X chromosomes are affected. Now, for a female to be born, you need two X chromosomes, one from the father, one from the mother. You only think, whatever X chromosomes are going to come from the father, it is going to be XH only. From the mother, it can be normal, but from the father, it is going to be XH only. And if you have one affected chromosome over here, since it's a dominant disorder, even one is enough. Okay, if one chromosome is there, obviously this female is going to be diseased. That is why I said you can see father to daughter transmission out there. And that is exactly what is happening up over here. So similarly, if at all you want the explanation or basically the tricks to remember for all of this, let me know. I'll make a separate video on it altogether. Otherwise, this video is going to go too long. Got it? So father to daughter transmission, you can see, or crisscross pattern. So that is there for each of them. There is some tricks out there. If you want me to tell you, let me know. Now, question number 4 is basically identification and commenting, and identify one of the givens below. That is TS, transverse section of testis, transverse section of ovary, vertical section of the ovary showing female gametophyte, and vertical section of the blastula. So any of these 4 can be given to you, and you might be asked to identify it. And my dear children, you need to comment also, 2 points. If at all you give 2 points, you get 2 marks. If at all you are able to identify it well, you get 1 mark. Okay, so over here the picture goes like this. This is the TS of the testis. This is basically a seminiferous tubule. Each of them are seminiferous tubules. Okay, and inside the seminiferous tubule, you know, Sertoli cells, sperm cells, everything is there. This is the human ovary. Okay, and over here this is the VS of an ovule, vertical section of an ovule. Now this is not exactly how in your book it will be there, or basically in your practical record or in your school it will be there. Please ask your teachers if you have any confusions to show you at least one, so that you have an idea. Now this is the blastula's image as well. Okay, so obviously have an idea of each of it. It is definitely going to help you. So blastula has inner cell mass and trophoblast, and this space is known as a blastocele. So there are some factors by which you can identify it, and please make sure you know about each of them, because writing about each of them also gives you marks. So if you're losing marks out there, it is going to be really sad news. So don't do that. Okay, study about them. At least 2 points each you can go. No, study and go. It's easy. It's not difficult. It's easy. Fine? Fine. Now there is one more question, that is second last question. Identification on any one of the given below, that is Ascaris lumbricoids, Entamoeba histolytica, Plasmodium vivax, ringworm fungus. So any of their image can be given to you. Again, you need to know their image. How do they look? Just go and understand it. Use Google. Okay, or as I said, if at all you want me to help, you can ask me also. So basically there will be slides shown to you. You have to identify them, and you have to put any 2 points about them. Half marks each would be given to each of these points. Half marks each. That means from the comments you will get 1 mark. From the identification you are going to get 1 mark. Okay, so write the name, what is the disease that they cause, and things like that. You can just talk about it, how is it spread. So yeah, that will give you marks. So have that common points for all. What is the disease that they cause and how it is spread. Have that, that's all. That would be enough. Now, next thing, question number 6 is identification of any one of the given below, that is controlled pollination, emasculation, tagging and bagging. So emasculation means you remove the male part from the female part, and then basically you do bagging. Okay, you do bagging, you cover this up so that no pollen grains comes over here. You are basically doing artificial hybridization. You are creating your own plants with the help of your own selected pollen grains out there, and then you tag it. So please know, they might give you a snip of it, they might show you a picture of it, some flashcards of it. So you need to comment on it, you need to identify it, and also you need to comment. For you need to write 2 points, half marks for each of the 2 points. That means 1 here, 1 mark over there. Okay, similarly, there's homologous and analogous organs. Know about each of them. Know the examples you study in your PU2. Okay, examples in the case of plants, examples in the case of animals. So thorns of tendrils, thorns of bougainvillea and tendrils of cucurbita, my dear children, they are homologous organs in the case of plants. In the case of animals, you have what do you call the limbs in the case of whale, cheetah, all the mammals out there, they are homologous organs. So know their pictures, it is there in your NCERT, just go through the picture, have it in your head and understand, okay, this is homologous organ, I have to write homologous. Okay, or these are the examples of analogous organs. So this is all attached to your theory part. So if your theory is good, you can write all the answers up here really easily. You can identify also really easily. Okay, done? So I think with that, you are partially ready. I know you are not fully ready because you need some experimental videos. And as I said, if you want, put it down. And my dear children, if you think that this video gave you clarity on at least few things related to your practicals, let me know about that also in the comment section below. Okay? And lots of love to you, and do not worry about anything. Okay? We are here. Yeah? See you soon. Bye-bye. Take care.